So in this video we will study the fourth property of the colligative property of solution, which is called osmosis. And osmosis, what is the definition of osmosis? So some substances form semi permeable membrane allowing some smaller particles to pass through but blocking the other particles. Semi permeable membrane is a kind of membrane which allow to something to pass, some small particle to pass but not some larger particle. And in biological system, most of the semi permeable membrane allow water to pass, but solutes are not free to do so, right? So this is the osmosis. Let's see in the picture form. So here is the this is the glass tube source, and here is a barrier between these two tube, which is a semi permeable membrane. This side is pure solvent water, and this side is solution, and which contains solute and solvent. So in case of semi permeable membrane, it allow only pass to water molecule from left to right side. So here you can see blue balls represent the water or solvent. They can pass this semi permeable membrane but not the solute. These green particles cannot go right to left. So after some time you can see the level of the water has decreases from this side, but the solution level increases, right? If you will allow some pressure, on the right hand side to make it equal level on both side, and the pressure, that pressure you can measure it. Basically that pressure, if you will measure, that will be called osmotic pressure, right? So in osmosis there is a net movement of solvent, basically solvent move from the area of higher solvent concentration, right? So this side is higher solvent concentration, low, or we can say lower solute concentration. So this side there is no solute or very less amount of solute, to the area of lower solvent concentration. So solvent move from higher solvent concentration to the lower solvent concentration, right? So this is the osmotic pressure definition or osmosis definition. Let's see how we calculate the osmotic pressure. So osmotic pressure was calculated through this equation. The pressure required to stop osmosis known as osmotic pressure, and it is represented by pi. Pi is equal to, in parenthesis, small n over V, basically number of moles over solvent. Then number of moles over solvent is molarity, and RT, R is a gas constant, T is temperature. So we can rearrange this equation like mRT. M is the molarity, R is a constant, and T is temperature, where M is the molarity of the solution. And if the osmotic pressure is the same on both sides of the membrane, that is the concentration of this are the same, means solution is isotonic. Isotonic means solvent and basically two side the solution have same concentration. That is called the isotonic. Let's see some example. Osmosis in the blood cells. So if a solute concentration outside the cell is greater than the inside the cell, the solution is called hypertonic, right? So suppose you place blood cells in the some kind of solution, and that outside solution have higher amount of, if the solution concentration is outside, if the cell is greater, higher amount of the solution is outside, more concentrated solution is outside. So basically what will happen, our cells loses the water, and the cell membrane is a kind of behave like a semi permeable membrane. So it allow water to come out because outside is less water or high concentration of solute. So the outside solution is called hypertonic solution, right? Because it have more solute concentration. So water will flow out the cell, and prenation will result, means cell will start dying. So another example of the Another terminology related to osmosis is osmosis in the cell. If a cell is placed in a solution where solute have less concentration, right? So what will happen? Solvent will go from outside to inside and cell will become, volume will become very high. So if the solute concentration outside the cell is less than that inside the cell, means outside the cell we have less solute, inside we have more solute, then what happen? Solvent comes from outside to in through this cell semi-permeable membrane. In that case, outside solution is called hypotonic because it have less osmotic pressure, less solute. So water will flow into the cell and hemolysis will result. Hemolysis means cell could burst because of the too much water. So these were the about the osmosis and colligative properties. Just go review a few more slides. So colloids, what are the colloids? Suspension of the particles larger than the individual ions or molecules but too small to be settled out by gravity are called colloids. So colloids are the examples. In this table they have shown some example of colloid: gas, liquid, and so colloid could be in any different solute and solvent can make colloid. And what is the definition? Basically they cannot suspend, they will remain in a suspension form in the atmosphere, they do not settle, they form the colloid. Next slide is this Tyndall effect. We have seen this slide earlier in the slides. Colloidal suspension can scatter rays of light, right? So any colloid, like in environment, they can scatter the light. This phenomenon is known as the Tyndall effect. And colloids in the biological system, some molecules have the polar hydrophilic water loving end and a non-polar hydrophobic water living ends. For an example, our protein have two ends. Sometime they have different, these are the red and blue, black and blue ball, shows that they have hydrophilic, means water loving group. For an example, OH group, right? COOH group, these are called hydrophilic water groups. So they, hydro means water, philic means loving, so they love the water. And the white part is shows the hydrophobic part or non-polar part. It like can dissolve in the fat or in oil, right? So this also, protein also form some colloids in the water. And another example is colloid in the biological system is like these molecules can aid in the emulsification of fat or oil in aqueous solution. So some molecule which have very large non-polar group and very small end which is polar, so they also form kind of micelle. Inside they will be all hydrophobic part, outside will be hydrophilic part, so they form the colloids. So this was about the chapter number 13, properties of solution. Please review all the slides very carefully, revise, understand the terminology, how to explain different words and how to solve the questions. Unit conversion is very basic part and very universal, everywhere you have to use those ones, so you have to memorize what are the units and how to convert those one. Okay, and if you have any question, please feel free to write question. I will try to explain or give you answer as soon as possible. Thank you.